Thank you, George, for the introduction. Thank you, everyone, for joining us today for this webinar. It is exciting, actually, to have so many participants taking off some time from your busy schedule. So the COVID-19 pandemic has taken the world by storm, spreading quickly across countries and causing an unprecedented number of casualties in a short period of time. According to the CDC, the last pandemic that most of us have witnessed, the 2009 H1N1 influenza virus pandemic, is estimated to be associated with possibly up to 500,000 deaths worldwide during the first year it circulated. It continues to circulate to this day, and it was deemed to be the worst in 40 years. However, to date, and with COVID-19, and in just 4 months, almost 2 million cases of the virus have been confirmed worldwide, with over 123,000 reported deaths. Here in the US, the numbers are around 600,000 or so reported cases, with almost 25,000 deaths. Unfortunately, these numbers are likely just underestimates of the true numbers of affected people and death linked to the disease. And despite the many advances in science and technology across many fields, this pandemic has uncovered how fragile and insufficient the processes and preparedness measures to tackle the emergence of a novel virulent virus are. Most of us are under lockdown at this point, while we're waiting impatiently for the flattening of the curve. The human toll has been exorbitant, yet sadly, perhaps, at least partially, it could have been avoidable. And the taxing economic price remains to be determined along with the future uncertainty it brings along. As we hear about opening up the economy, we need to plan for a phased approach, coupled to metrics and the implementation of a solid surveillance plan to provide us with the needed confidence to return to a new normal that has yet to be defined. And as we prepare and organize ourselves for another possible pandemic wave in the course of the year, we look to science to hopefully deliver a needed timely solution. It is therefore my pleasure today to host 3 experts who have been associated with the current COVID-19 pandemic in various capacity and have graciously accepted to be part of this LebNet event to share with us their experience and insight from the front line to finding solutions and cure. Dr. Salwan AbiEzzi will give us an overview of the spread of the disease and what types of measures need to be in place before resuming a semblance of normalcy. Mr. Ammar Qadan will share with us how Illumina, the pioneering genomics company, is capitalizing on their technology to expedite our understanding of the virus and offer tools to establish solid surveillance programs. Last but not least, Mr. Muhieddine Makkouk will bring us up to speed on the ongoing global scientific race to develop and deliver drugs to cure and or prevent the disease. I should add that the very popular topic of testing that we hear constantly about on the news will not be covered today, but if there are a few questions between the 4 of us, we could attempt to answer them at the end. Salwan will lead the way with this overview, but prior to that, I would like to make the proper introductions. Salwan is lead physician at the Palo Alto Medical Foundation, where he practices general internal medicine with special interest in diabetes and sports medicine since 1999. Salwan majored in biology, but also with 2 minors in human psychology and computer science, graduating magna cum laude from Rensselaer Polytechnic Institute in New York. He obtained his MD degree from the University of California, San Diego, and he completed a 2-year postdoctoral research fellowship in orthopedic surgery. He then went on to complete his internship at the University of California in San Francisco and his residency in internal medicine at Santa Clara Valley Medical Center in San Jose, and additional training at Stanford hospitals. Without further ado, Salwan, the floor, I guess the screen is yours. Thank you.
Thank you, Farah. Let me pull up my slides. Oops. Just give me one sec. Insert slideshow from the beginning. Can you see my slides?
No. Oh, it's coming. It's coming. Yes, now we can.
Okay, thank you. So thank you, Farah. Thank you for this introduction. Well, I'd like to focus today as a clinician on three aspects of this COVID-19 pandemic. One is what's the nature of a pandemic and the epidemiology of a pandemic. The second is the clinical manifestations as a clinician in terms of symptoms. And the third, how we're gonna imagine life coming back to normal going forward, the public health aspect of it. I've used some of my data from UCSF medicine grand rounds and Stanford medicine grand rounds that we've had streamed recently. So I want to remind everybody with the slide as to why this is such an important event in our lives and why we're making such a big deal about this and why we are all sheltering in place today. We have an experience like this in the last 100 years, which is a Spanish flu or the H1N1 influenza pandemic of 1918. And this slide captures actually how infectious diseases actually cause a lot of death and suffering in humanity until maybe the last 75 years when we've invented better public health measures, antibiotics. But you see this massive spike, 1918, this is a H1N1 influenza pandemic, which caused the death of about 50 million people and infected about 500 million people, which is about the quarter of the population on Earth at that time. So imagine, and it's by far the deadliest pandemic. Now, pandemics, by definition, are infections without borders. This is coming from Dr. George Rutherford, who's a premier professor of epidemiology at UCSF. And so by definition, a pandemic is a type of an infection that is highly virulent and crosses borders and communities and geographic barriers fairly easily and can spread logarithmically. And you can see from the slide early on, from the early reports out of China in end of December, early January, we saw reports coming from South Korea, Iran, and Italy. And we were able to trace those to, for example, South Korea was a church group that brought it in from China. For Iran, it was actually 600 pilgrims that went from Wuhan into Qom, which is a pilgrimage site, a Shia pilgrimage site. And then obviously in Italy, because of Venice and Milan being a central hub for tourism and industry in Europe. And we have also the example of the Diamond Princess ship, which is a perfect example of how in a contained space, the virus infected a number of people in a logarithmic scale. So if you look at the rest of the world in terms of number of infections, you'll see this kind of a bump in China, but China having had experience with SARS-CoV-1, which is a cousin of our current SARS-CoV-2, they have better preparedness. They were able to lock down that entire region and kind of arrest the pandemic, but then we saw it spreading. If you look at the orange graph, that's kind of Europe, Eastern Mediterranean is splashed on top of that, and then the United States and the Americas up on top, which is now basically the hotspots of the pandemic. And down here below, you see the southern regions of the world that have not been affected yet, which are Africa, for example, and Australia. This is you're familiar with this map. This is from 2 days ago. We already surpassed 600,000 confirmed cases of COVID-19 and more than 30,000 deaths, unfortunately, here in the United States. And you see a huge discrepancy between the West and the East Coast. And obviously, there are many manufacturers that come into play. But the tri-states of New York, Pennsylvania, and Connecticut are the hotspot right now in the United States. Initially coming out of Wuhan, we thought it was predominantly affecting older adults, and the majority of people that died were older adults. But data that came to us more recently in the last from February 12 to March 16 here in the United States suggest that all age groups are affected by this virus. The majority tend to be young adults and older adults, which are in the age of 20 to 70, 75, but the majority of the people that die tend to be older adults or people with comorbidities. Again, I'm gonna go back to the Spanish influenza pandemic of 1918 to show this great slide, also courtesy of Dr. George Rutherford, basically showing that there were two cities in the United States that adopted different measures. One was St. Louis, where the pandemic originated. They adopted a shelter-in-place policy very early on, and Philadelphia did not. And you saw kind of a massive upsurge in deaths in, this is in terms of thousands per 100,000 population in Philadelphia. It was flattened early in St. Louis. What was interesting in that pandemic, which lasted about a year, it started in January actually, in 2018, I believe, and then went to December, is that there was a slight uptick of the virus causing deaths in St. Louis towards the fall. The key message from that pandemic is that there were these closures that happened very early. Closures included theaters, moving picture shows, schools, pool and billiard halls, Sunday schools, cabaret, lodges, societies, public funerals, open air meetings, dance halls, and conventions until further notice. So the key message from that lesson is begin social distancing early and keep them going throughout the outbreak period. So to take this further, we have, and this is data that came to us from UCSF. These were undergraduate and graduate students that were put to the task in the epidemiology department to look at data from these two regions of Italy that were adjacent to each other. One was Lodi, which is adopted a shelter-in-place ordinance earlier than Bergamo, and you can see the difference in terms of mortality between those two regions. Reaffirming the power of the shelter-in-place ordinance we're in today. Another set of data presented also suggests this point is comparing San Francisco to Los Angeles. San Francisco adopted that shelter-in-place order a few days before Los Angeles, and there was a significant difference between those two cities. Additional data, which was quite interesting, came also, this is again from UCSF, where they looked at open table reservations and compared three cities, San Francisco Bay Area, Los Angeles, and New York. And you can see the open table reservations dropped much more rapidly in San Francisco, trailing behind were New York and LA. And we had many Bay Area Silicon Valley companies actually recommend and begin telecommuting earlier than the ordinance of March 16. Salesforce March 4th, Apple March 6th, Google March 10th, Twitter March 11th, and actually UCSF was also early March 13th. And I remember the Catholic schools, I have 4 children, one of them was going to a Catholic school, they shut down the schools a week before the public school system. So people were a little bit ahead of the curve here in the Bay Area, and we are benefiting from that. So this is a slide that captures the COVID-19 nasopharyngeal tests that were done on our moderate severe symptomatic patients in the Sutter Bay Medical Foundation, which includes 4 large hospitals around the Bay Area, including CPMC in the city, California Pacific, and Mills Peninsula Hospital in Burlingame. And you could see that early on, we were getting a significant surge in the number of positive cases, and that reached about 10%, which is like 40 out of 400. And that kind of maintained through the mid level of March. But in the last week, I have to say that we flattened the curve. We are having days where we're seeing no positive COVID-19 cases out of the many that are being tested with respiratory symptoms in our regional hospitals here. So this is the good news here in the Bay Area. George asked me to give a kind of a quick presentation on the symptoms and how, as a citizen, you can distinguish non-severe from severe symptoms. And for this virus, it turns out about 80% of people who contract this virus will have non-severe symptoms, and about 20% will have severe symptoms, moderate to severe symptoms. 5% of them will require intensive care units. So this is a highly, highly virulent virus. The main symptoms that we are all aware of now include fever in about 70% of the time. However, there are many patients that get admitted without a fever and then develop the fever in the hospital. Cough, which is predominantly a dry cough. It's a cough that emanates more from the chest, and it's dry, it's not phlegmy. It's not the rhinovirus type cough where you have a lot of nasal congestion and the cough is emanating more from the throat. This is a deeper cough. A subset of patients will develop shortness of breath, but not the majority. It tends to have flu-like symptoms of fatigue, myalgia, which means muscle pain, joint aches, as if you've been hit by a truck. That's a common symptom. Another common symptom that came out later is anorexia, which means low appetite. Very common, 80% of people had a low appetite. So if you have a cold, but you're eating your heart out, it's probably not COVID-19. It turns out that some people have upper respiratory symptoms, including sore throat, nasal congestion, drainage, headaches, and some will have gastrointestinal symptoms like nausea, vomiting, diarrhea, but those tend to be rare. More recent reports of losing your taste for a period of time and so forth. So ultimately, why there are a few that get really, really sick? There are many factors that contribute. Obviously, the older the person, the more comorbidities the person has. Obesity came out to be a very, very serious comorbidity. Diabetes, hypertension, male, a little more than female. We still don't understand the reasons behind that. But it's a lot of times what we know from influenza and SARS-CoV-1, it's the inflammatory immune response that can lead to a lot of the damage to the lung, predominantly the lung alveoli. That's the main organ system that gets damaged, which is your air sacs in your lungs. And I remember one ICU attending told me 4 weeks ago, I had a patient admitted and is in the ICU, that this is a uni-organ disease. That's the main organ system that's affected is the lungs. Usually people might have slight stress on their kidney or liver or the brain, and like they might get a little confused. However, the main organ system that gets damaged is the lungs. So people tend to have this shortness of breath, cough, and eventually, because the oxygen level drops, they get confused and lethargic. So what are the antidotes for the common citizen? Is to really, first of all, take care of yourself. So I have a mantra that I've been preaching for the last 10 years here at PANF, and I'm sure some of you have heard my lecture. I call it the 4 pillars of healthy living. And there's a ton of data on influenza that if you eat right, you're less likely to get the flu. And I want to stress there, the food that tends to be calorie dense, but nutrient poor is not likely to be good for you. And alcohol specifically weakens the immune system. So even though my wife Farah mentioned yesterday that the alcohol consumption went up 50% since the shelter in place, I don't think that's a good idea to overdrink during this period of time. So focus on healthy nutrition. Us Lebanese, we know what that is. Lebanese food, Mediterranean food, close to the earth food. Exercise or activity is critical. People who exercise have a stronger immune system, less likely to get influenza, less likely to die from influenza. So it boosts your immune system. I want to highlight with this slide being outdoors, because there's also a lot of evidence that vitamin D, which you get from the sun, also improves your immune system. People who tend to be vitamin D deficient are more likely to develop influenza or die from it. Taking supplementation for the folks who are not able to get the sun is also beneficial. So in this time period, I would recommend that for people who are not able to get the sun, but that's the most effective way to get vitamin D. Sleep is critical. Again, there's a lot of research that suggests that people who get a good night's sleep have a stronger immune system, better metabolic system and less likely to be sick from influenza. So a lot of this data is on influenza, but it kind of, the influenza and SARS-CoV-2 are both riboviruses, RNA viruses, so behave the same or similar in terms of the immune response. And the fourth pillar I would like to focus on is a healthy mindset. We talk about relationships as a foundation for that, open mindset, being accepting, being resilient. Us Lebanese, we know what it's like to be resilient because we faced adversity in the past. We are all experiencing some grief right now. Some people might have it as anger or fear or anxiety, depression. Those, unfortunately, mental stressors will increase after a pandemic. It might take a year or two to resolve. So the key advice I would give you there is to really reach out and connect virtually because we are social beings, we're human, and especially Lebanese, we love to connect, we love to hug, we kiss, we want to gather. Right now, you only can do it virtually. So George also wanted me to give like tidbits, practical tidbits. I really reduced it to those 4 slides. Wash your hands, wash your hands. And if you can't wash your hands, use a 70% alcohol gel. That's critical because the virus is transmitted by droplet form and tends to hang out on surfaces, and you touch these surfaces and you touch your mouth or your eyes or nose and you bring the virus into your body. So washing your hands often is key. The other is wearing a mask. I think as a society here in the United States, we have not adopted that yet. China is way ahead of us, again, because they've experienced pandemics in the recent past, like SARS-CoV-1. They've had the Hong Kong pandemic, that was 1967. H1N1 originated in China, that was 11 years ago. So they're really comfortable social distancing, keeping a distance away from each other when you're sitting on a bus or a train, and wearing the mask. The mask predominantly keeps your fomites, your saliva within you, so you're less likely to be infectious. That's the main benefit of the mask. Also reminds you not to touch your mouth. Cleaning surfaces is key. So I see people sometimes go overboard with spraying Lysol on their produce, and the benefit of doing that is probably marginal. Most of the infections that have happened in China and in the world happen from close contact, having a member in your own family who's sick. So you have to have a high load of the virus. You have to be in close proximity to these droplets. So I don't know how much of that transmission is going to happen from a carton of milk, for example, that you're getting from the store.
Dr. Sal, sorry, this is George. We're kind of running out of time, so just calling time here for.
Okay, thank you. I'm almost done. So having some medication such as Tylenol, Advil, there's controversy about using Advil, cough syrup, having a thermometer at home. Some people like to have oxygen monitors and so forth. I'm going to end with these 2 slides that come again from Dr. Rutherford in epidemiology. So we know that we need to minimize the transmission of SARS-CoV-2. We need to avoid overwhelming the medical care system. That's why we did this shelter in place, and we don't want to be like New York. And we all want to return to life we aspire to, and then contain this transmission until we have a vaccine. These were the four main goals. And I want to end with this busy slide, but I'll go through it, George, very quickly. We need to, before we remove that shelter in place, we need to show that transmission has become low. Low numbers of new cases, low numbers of hospital admissions, low cohort or asymptomatic patients in the community. We can do surveillance through many ways, including like Kinsa, which is a thermometer company that kind of tracks fevers. Citizen scientists. We need to have the test be available fairly essentially rapidly and on demand. Google is partnering with Verily to provide that to people who don't have insurance. We need to have a vigorous contact tracing, supervision, isolation in place, and the ability to quarantine people. We need to have an adequate staffing of ICUs and hospitals and ventilator capacity. And then we need to all agree on social distancing, sheltering in place. For example, older adults or vulnerable people. Options for schools, masks should be worn more readily, and then staggering return to work, such as manufacturing and certain industries before others. And I'd like to end with that. Thank you.
Thank you, Salwan. for the great overview. And basically, it is obvious from what you mentioned to us that obviously shelter in place is critical, certainly in this early phase of pandemic. But as we brace and we prepare ourselves to get back, and we hear so much about opening up the economy, as you mentioned, the surveillance program is truly a must.
Yes.
And Ammar will next walk us through some of the technical advances that have been made that hopefully are going to help us basically get to that place where we would have surveillance programs that are coupled with testing. So I would introduce Ammar again. So Ammar is currently Vice President and Global Head of Market Access at Illumina. Ammar is in his job and his current role, Ammar is responsible for health economics and outcomes research, market access, pricing strategy, field market access, payer policy, and advocacy. Throughout his career, Ammar has been a distinguished and passionate and innovative, patient-focused leader who worked in different healthcare industries, multiple global geographies, multiple disease areas, products in different life cycle stages, including immuno-oncology, virology, cardiovascular diseases, virology, and diabetes. At Illumina, Ammar built the market access function, designed and executed the first value-based contract in next generation sequencing that he will talk to us about. And prior to Illumina, Ammar was the Vice President and Global Product Team Lead at Allogene, where he basically led several programs and managed partnerships with Roche Genentech. And prior to joining Allogene, Ammar spent nearly 20 years at Bristol Myers. Ammar holds a degree in pharmacy. And Ammar, please, the floor is yours.
Thank you, Farah, and thanks, George. And special shout also to Layal Rohana, who coordinated with me initially. I wanted to go quickly through some of the basic concepts for those who are non-scientists on the call, but I will go quickly around the DNA basics. So we know that DNA has 4 chemical groups, and those chemical groups, they bind together in a certain way. However, also it's important to mention that the sequence of those chemical groups, or what we call the nucleotides or the bases, is important. When we do sequencing, really we cut the DNA into smaller fragments, and then through bioinformatics and other capabilities, we will be able to read the genome or part of the genome as required. It's important to mention that the human genome has 3 billion of those bases, while the virus, which is an RNA rather than a DNA, it's just single-stranded, has 30,000 bases. So it's easier definitely to read the virus than reading the human genome. But if you're looking at the whole genome, whether it is the virus or the human being, it's called whole genome sequencing. So it's important to mention all of that because as we talk also about the mutations that viruses and diseases also, like cancers, go through, it's important to understand that these could be different types of mutations. And the most common probably is what's called single nucleotide variant, whereby just one base is changed. But there are other types of mutations, as I said, including what's called insertion deletion variants, where a whole block of bases is taken out, basically is deleted, or sometimes they refer to as endals. These 2 types of mutations is what we have seen with the coronavirus. There are other types of mutations you can see here. Towards the end, you can see copy number variants. This is most common probably in rare diseases, for example. And I'll talk about some of these clinical applications in a minute. Everybody talks about the previous pandemics, including the 2014 Ebola, the 2009 H1N1, but I can tell you that we're dealing now with those pandemics with a technology that did not almost exist with previous pandemics. To give you just an example of the scale of technology evolution that we're talking about here, when the first human genome project was finalized in 2003, the cost of that program was around $3 billion. It took 13 years to finalize the first human genome. Today, we're talking about probably less than 24 hours and for less than $1,000. For those who are coming from a technology background, they understand and they can relate exactly to what I'm talking about here in terms of the technology development. And proudly saying this has been done really mostly with the technology development that Illumina, my current company, is leading. So what's the value of all of that? It has opened huge clinical applications. There are clinical applications, there are research applications, but there are also applications in agriculture, for example. And let me highlight some of those applications quickly for people just to understand what we're talking about. In reproductive health, for example, before next generation sequencing was introduced and before it was scaled up as we see it today, the standard of care for a pregnant woman within the first trimester was serum screening. And if a woman goes and does a serum screening and turns to be there is something wrong with the baby, there is some chromosomal abnormality, the next test to be done is really an invasive test where they take samples from the amniotic fluid. And we know that this is associated with a risk of abortion. That test, the serum screening, is associated with almost 5% false positive rate. The next generation sequencing, you can do a blood draw from a pregnant woman called non-invasive prenatal testing, and the false positive rate is 0.05%. So you can think about how many invasive tests you could be saving just because of doing something like this, and perhaps also saving abortions. The other area that is growing fast is oncology. I remember when I worked in oncology 10, 15 years ago, there were probably 2 or 3 drugs with the biomarker called biomarker-driven program. Today in oncology, all drugs under FDA review or that have been launched already, they are biomarker-driven. And in many of those cases, you need also next generation sequencing. The most development I was reading today, for example, is the blood test now that is being utilized in oncology called liquid biopsy. And so basically, traditionally in lung cancer, you need to have a tissue from the lung. Now you can take a blood draw and would be able to do exactly what you used to do on the tissue and identify really the biomarker associated with that cancer. In addition, some of the recent data, which was released yesterday and today, this would enable you also to be able to look at relapse of cancer 6 months earlier probably than what you get with something like a PET scan. So imagine now the evolution in that field. The other area I would like to focus on is genetic health. These are the rare diseases, for example. And we have a lot in the Arab world with rare diseases for obvious reasons. But I can tell you traditionally this has taken probably 7 years to diagnose a rare disease, 7 whole years. And the kids, they go through different types of tests. Today, there is a hospital here in San Diego, and there are many other centers around the nation, and also in Europe and many other countries. You can diagnose those patients in 24 hours. There is a children's hospital here in San Diego called Rady Children's Hospital. That's the degree of development that we're talking about. In addition, there are other applications like consumer, 23andMe and others as well. So what type of technology you need to use in order to diagnose those types of mutations and so on? Like the case with coronavirus, you could be using what's to the left here, the PCR, the polymerase chain reaction. If you know what you're looking for, you can really target that specific mutation and just do the test with something like PCR, which is less sophisticated. It's more cumbersome. But if you do not know what you're dealing with, you need to do some kind of genome sequencing or even whole genome sequencing. The issues that we're dealing with today with coronavirus is that if you want to develop a test like PCR to look for the virus, you need to understand the genetic information of the virus, and that can only be done with next generation sequencing. So you can see the correlation between the two areas. Something like rare diseases, you need to do whole genome sequencing. You cannot really rely on PCR. So what's going on today based on what Salwan and Farah mentioned around surveillance and diagnosis? In fact, you could use really next generation sequencing for diagnosis also of the coronavirus. But as I said, if you know what you're looking for, maybe you can use a cheaper test like PCR. However, in terms of surveillance, surveillance is very important for different reasons. I'll mention those reasons quickly in a minute. But what we do with surveillance or with testing, sometimes you do the test and you do also next generation sequencing for the virus of that sample. And then you store all of those in global databases so that it is available for others to look at. And that's exactly what happened when we started seeing the cases with coronavirus. We go back to those databases and look at similarities. And that's how we were able to diagnose coronavirus. In addition, next generation sequencing, of course, I mean, it could have much more sensitivity in terms of testing. And that is important, especially in early stages when the patient is not symptomatic. I'll talk about that maybe briefly. But let me talk a little bit more around surveillance because that's really interesting. So what are the uses of surveillance? The first thing is identifying the virus. So that's exactly what happened with when the first outbreak happened in Wuhan in China. They were using really our platforms. And what happens usually is that you do what's called shotgun sequencing. You do not know what you're looking for. And all what you do is get all the genetic materials from that sample and start looking at, is this a genetic material of the host, the human being? Is this a genetic material of something else? And what that could be. And then you start looking at those genetic materials that are not common, like the coronavirus, and you go back to the databases and compare your sequencing results with other available resources, open resources. And that's very important in defining any new pandemic or outbreak. That happened exactly with Ebola virus as well, where we shipped one of our sequencers to Africa. They did also shotgun sequencing, and they were able to identify really the sequencing of the Ebola virus. But I can tell you that process in 2014, when that happened, could have taken probably weeks or months. Now, as I said, it takes hours or just a day or two. The second thing in surveillance is tracking the transmission route. For example, we know the first cases were in Washington state. So Washington University quickly, they went ahead and sequenced the samples coming from those patients. 39 samples were sequenced quickly. 35 of those could be traced back to Wuhan, to the original virus. 3 were traced back to Europe, and 1 was traced back to Iran. And so that was really when everybody started thinking that this is not coming only from one place. So if you want to shut the flights from China, you're not doing everything. And that stimulated really the discussion about lockdown. So that's very important. In addition, when you look at the third point, which is monitoring the viral adaptation, so the virus is mutating almost 2 mutations per month. If you correlate that with the time when the sample was taken, you will be able to exactly see how the virus evolved over time and how long has the virus been in the community. And that tells you basically whether you need to take much more stringent, or drastic measures or not. And that's also the major value of doing next generation sequencing, again, because you will be able to find some genetic material from perhaps future outbreaks. The fourth important point is identifying vaccine and therapeutic targets. For example, what we know about the coronavirus is that it shares some characteristics, and I know Muhieddine will talk about the drugs in a minute. It shares some characteristics around the protease enzymes with some of the initially used antiviral therapies in SARS and other types of viral infections. Almost 96%, they have common targets. When it comes to the vaccine, those that are really the parts of the virus that are needed for development of the vaccine, there was a similarity of previous viruses of around 76%. So what that tells you is that perhaps previous treatments could work here, but current vaccines cannot work here. You need to have much more similarities with the targets with the virus. Last but not least is understanding the role of co-infection. So some of those patients had really, the coronavirus was morbid, some were not. That now is being studied. So they take also the human DNA and start looking at some correlations between the outcomes as well as the human DNA, trying to find why some patients survived while others not. But also it's important to do next generation sequencing because you might be able to find a bacterial co-infection rather than just the coronavirus. And that could be the reason behind the mortality in those patients. So I'll close with two slides quickly. What we're talking about here in terms of having a surveillance program has really not only the values that we talked about, but you can translate every single one of those values into societal value, whether it is avoiding community transmission in asymptomatic or mildly ill patients. And the cost of that is almost $8 billion in the US. Whether it is better treatment, stability of families, vaccine development, vaccine effectiveness, so that currently we do not see the impact of mutations on the need for changing the vaccine of for the virus under development. But what they have seen so far is that when they go and inject those cells into the monkey cells, which they do in order to produce on commercial scale, they notice that the pattern in monkey cells changes quickly and you have different types of mutations. And that has a huge impact on the development of the vaccine. The last one is again, potential to inform longer term therapy development. I know Muhieddine will talk about those types of things, but all of those things could be quantified as well in terms of money, so that when we talk about the economy, it's not only the loss that we see today, but the loss, but the fact that you could prevent some of that or reduce some of that. Last but not least, Illumina was really on the forefront with the University of Washington. We have done the work with them around the sequencing. The Diamond Princess, in Japan, we gave them our machines, and so they sequenced all the patients really on the ship. Currently, we have 5,000 genomes sequenced for the coronavirus. So we have a good number of genomes. Last but not least, there is also an embargo on Iran as we know it today, but Iran was one of the hardly hit countries. And so we asked the State Department, and we were able to ship some of our sequencers as well to Iran to deal with the pandemic there. So a lot of work going on. I can tell you this is something that is changing on a daily basis. And we have regular calls on a daily basis with some of the governments and some of also of those who own the open data sets looking for new information through some of the genomic sequencing that we're doing. So with that, I'll turn it back to you, Farah.
Thank you very much, Ammar, for this overview of this amazing and powerful technology, highlighting all those applications that can be useful. As you mentioned, the steps of implementing surveillance programs are critical for the next step for us to be able to figure out how we get back to our normal life. But then ultimately what is key is, will we have a cure or a vaccine on the horizon? And with that, I will let Muhieddine go ahead, but prior to that, let me just introduce him. Muhieddine is currently Vice President, Alliance Management and Integration at Gilead Sciences in Foster City. In this capacity, he oversees teams responsible for managing over 150 collaboration from early research, discovery, clinical development, and commercial. Muhieddine has also established and leads the Integration Management Office at Gilead. Prior to his current role, Muhieddine managed various commercial regions, including the EMA in Europe, distributor markets, EMEA region East, and the Gilead Nordic cluster. And prior to Gilead, Muhieddine assumed roles of increasing responsibility across multiple geographies, therapeutic areas, and function with Merck and Novartis. Muhieddine began his career at IMS Health and has over 27 years of pharma sector experience. He completed his graduate studies in health policies and health economics. Muhieddine, your turn. No pressure, but I think hopefully you will be able to cover things in 20 minutes.
Yeah, we'll try to whiz through. Thanks, Farah, for the introduction. I think, there's a lot of news that's out there. I think many of those on the webinar have been hearing about, hopeful sort of glimmers of hope that products are on the horizon. But I think we need to manage expectations and probably shed a little bit of light on the both risky and lengthy, development cycle for drugs. With that, let me actually go to our first slide. So, we've been hearing about treatments as well as the vaccine. Those are really the two interventions that will help manage this pandemic. Of course, the treatments are key because they will help us not only improve outcomes for patients, reduce mortality, but also reduce the length of hospital stays potentially and the pressure on the healthcare system. But ultimately, we won't really be able to tackle this until we have a vaccine, which is probably a little bit further out. And talking about really the lengthy R&D development for drugs, I mean, in an ideal situation, this is a cycle of 7 to 10 years, which we're really trying to compress. But there are only so many corners that we can cut and still have a safe and efficacious a drug that can be used across populations. Not only that, of course, there is a very complex process which goes into manufacturing for vaccines, and all of that has to be compressed to make a treatment available quickly. There's a lot of good news out there, and we need to be optimistic, but I think we need to be cautiously optimistic. If we start looking at the numbers, I mean, there's roughly close to 250 different programs out there, between 160 to 70 therapeutics. And I'm sure these numbers are already incorrect because there's daily news of new programs and targets that are being explored, and over 70 vaccines in development. But unfortunately, most of these are in very early stages. If you look at the number of trials, there's over 250 clinical trials. Doesn't mean these are pertaining to specific targets. Many of them are in combinations and for some products that are already available on the market. But also quite a gigantic task running those trials and really getting them off the ground so quickly. But if we look at the odds, I mean, the odds aren't that bad. I mean, for infectious disease treatments, they're 1 in 5. So a compound that enters clinical development has 20% chance of making it to approval. So that in itself is encouraging. And, ultimately what is the most encouraging is we have so many shots on goal. So with these numbers, the likelihood is ever increasing that there will be a treatment and hopefully a vaccine as well in the not so distant future. We're also lucky that we know how the coronavirus structure and how it behaves. If it was an unknown family, of course, this would have made the whole process much more challenging. And one key point to also sort of shed light on is the fact that not only do researchers and companies understand the urgency, but regulators are becoming much more pragmatic in dialoguing with the industry to ensure that, if something is starting to prove, to be efficacious and safe, that there will be accelerated timelines for review. Gilead, as well as many others, have been in very close, almost daily dialogue with the FDA, the European Medicines Agency, and other national bodies in that regard. And this is really very heartwarming. The other, I think, dynamic that we see is that many centers, maybe Salwan can also speak to that, have sort of shifted some of their resources to basically COVID trials. So suspending trials on other medicines and creating a bit more capacity to accelerate the COVID trials on the different compounds that are available. So really a lot to be optimistic about, but of course, still a lot of hurdles ahead of us. And if I really wanted to sort of bucket, the efforts today, they really revolve around three areas, or we're really moving fast on three fronts. You have the first group, which is the current drugs. These are approved for other indications, bear in mind. And these are older medicines for malaria, for rheumatoid arthritis, and older antivirals. You have the whole new sort of group of antivirals and monoclonal antibodies, and of course, new vaccines, as Ammar said. While some of the older drugs may prove useful, with vaccines, I think this is a whole new attempt, and we'll talk a little bit more about that in the coming slides. Just very quickly, so if we looked at group one, the existing therapeutics, you've heard a lot about hydroxychloroquine and chloroquine. These are the malaria drugs. Unfortunately, there's only anecdotal evidence, very small trials in France and other places. We're starting to see these used in more broader protocols, but there's, again, no, I would say, conclusive data as of yet. Some of the trials or protocols are combining those with azithromycin, which is an antibiotic. We've also seen approval in Japan for an antiviral, but again, based on very small sets of data. We've seen more recently encouraging data for autoimmune drugs like Acterma and Kevzara, which are used for RA, rheumatoid arthritis. These can help potentially mitigate the cytokine storm or the immune response associated with COVID infection. There's some early encouraging data. We've already seen an approval in China. Roche, for example, is running broader US studies for Acterma as a standalone, but also in combination with antivirals. And you've also heard, and this is really not a drug, it's more of a blood derivative product, convalescent plasma. This is an old, as you know, approach, has proven effective in many cases. However, scaling that up is the challenge, matching donors, and really having a solution that is off the shelf is where the challenge is. So this is really sort of the landscape of what's available now and what's being explored in terms of currently approved therapeutics. Obviously, the second group is the new antivirals and antibodies. At the sort of the front of the race there is Remdesivir, which is a drug discovered by Gilead. The reason it's at the front of the race is it was discovered back in 2009, in fact, and it was developed originally not for Ebola, but actually for hepatitis C and for RSV, and was explored in those indications initially. But given its broad action, has also been explored for other unmet medical needs, such as Ebola, SARS, and MERS, and obviously now in COVID-19. I will share in a couple of slides some very recent encouraging data, but again, nothing conclusive. We've also seen use of antibody or a mix of antibodies that's proven effective in Ebola. Regeneron is working on a combination there, and also Eli Lilly and Accelera, as well as Biogen and Vir Biotechnology are developing a similar approach of a combination of antibodies. these are still, I mean, months, if not longer out, you know, into the future. And the third group is vaccines. You know, here the timelines really vary. I will share with you in a minute a slide from J&J. They are quite bullish on their timelines, where potentially in Q1 2021, we may see early use or emergency use of their vaccine, but this may take many, many years. Just the fact that we enter, you know, into a vaccine program on one end doesn't mean we'll come out with an approved vaccine on the other. So I think we need to be cautious here about that. But as I mentioned earlier, there are 70 vaccines in development as of yesterday, but only 3 in clinical trials. The most advanced is a vaccine being developed in China. That's entered phase 2. And there are 2 vaccines in the US that are currently in phase 1. The rest are all in preclinical trials at this stage. The other observation is that many of these efforts are done by smaller companies, smaller startups, and academic institutions. However, there are a few efforts which are underway by larger multinational vaccine developers. And the reason for this distinction is the ability of those smaller companies to bring a successful vaccine to market and to scale it up very rapidly. So the larger players are J&J, and we'll look at their timeline in a second, as well as Sanofi, GSK, and Pfizer. They all have programs that are underway. One specific observation as well in this space is that many of these programs are working on technologies that are not the basis for any currently licensed vaccine. So that obviously complicates, you know, the chances, but also the odds of success. And the industry benchmark in terms of, you know, attrition of vaccine programs is more than 90%. So, you know, the failure rate is quite high. But again, with this number of programs, you know, across the whole continuum, you know, the odds still are somewhat favorable, and we will likely need more than 1 vaccine. So, you know, we're on the right path. Looking actually at J&J's Q1 earnings just on Monday, so a few days ago, they shared with the public their program. And as you can see in the slide, they have established now a lead compound. However, they will not be able to enter into human trials or first in human study until September. So as you can see, we're still months away even from initiating human trials. And, you know, again, quite optimistic. Again, never been done before, but, you know, we can always break the rules as long as we have the right guardrails and we produce the right evidence, not only for efficacy, but safety. And we've seen, you know, vaccines in the past fail. We've seen a dengue vaccine, for example, with side effects emerging, for example, in the Philippines, well into its commercialization. So I think we need to be mindful of the risks also associated with these programs. So with that, what I would like to do is transition to what Gilead has been you know, doing in the last several months and actually years, but more recently on addressing COVID-19. To give you a little background on Remdesivir. So it's obviously still an investigational, very broad spectrum antiviral. I mentioned that it was initially developed to you know, assess its impact in HCV and RSV. We have been working on it in COVID-19 for, you know, almost since early January. There are multiple trials, and I'll share that in a slide. In fact, there are roughly 8 clinical trials, 5 where we have been very closely involved, and 3 that are being run globally by the WHO. To date, we have more than 1,800 patients that have received drug through the compassionate program, and that is outside of the clinical program. and we're activating an expanded access given the tremendous number of compassionate requests. At one point, the system just could not cope. Just before converting into an expanded access, we were getting over 100 requests an hour for compassionate use. So we've moved it into an expanded access program. Our approach, I think, as well as many larger companies, is really to work on 3 tracks in parallel. One is establishing the safety and efficacy of the target, and in this case, remdesivir. Scaling up manufacturing in parallel, and obviously at risk, as no one is certain if this or any other compound will be approved, and also working on delivering responsible access, based on the epidemiology. And as Salwan showed, we've seen the flattening of the curve in China, followed then by the uptake in Europe and the US, but also in the Southern Hemisphere, the numbers are quite low. All of this can change. So we need to make sure that we're not looking backwards, but looking forwards based on the emerging epidemiology to make sure that the drug is available where it's needed most. Looking at, again, the first sort of area of focus, which has been establishing safety and efficacy. This is just a quick overview of the trials. These are all phase 3 randomized trials. 2 of the trials are open label. The other 3 are randomized double blind. The trials in China, in fact, had many obstacles. And I think one of the issues there was the rush of many clinical trials into China almost at the same time, which put a burden on the system and obviously issues with trial startup. And we're starting, unfortunately, to see the results of that on the back end. However, the US and European trials are progressing very, very rapidly. We saw a few weeks ago I would say a report or a study published in the New England Journal of Medicine, which has been very encouraging. Again, it is a sample of patients. It is in no way conclusive, but data on 53 patients treated with remdesivir. The data is, as I said, very encouraging, with 68% of these patients showing clinical improvement. And the majority of patients did not demonstrate any new safety signals. So, I mean, the safety is clearly in line with what we've seen previously, and the clinical response has been again, encouraging. Again, this is anecdotal information or data, and until we have a broader readout of the trials, there will be, I would say, caution, but I mean, we're sharing the data as it evolves with the FDA, and as I mentioned, almost in daily discussions with regulators. In parallel, manufacturing scale-up is key. The process to manufacture remdesivir is quite lengthy. It's a very linear chemical synthesis process. It involves many substances and multiple suppliers across the globe. We've been able to cut the process down from roughly 12 months to just over 6 months, which is significant. And we've been ramping up production. We do expect to reach 140 treatment courses by the end of May. And Gilead has made the commitment to donate all of that free of charge. And we continue, again, at risk to scale up, hopefully reaching 1 million treatment courses by the end of the year, with the hope, obviously, that remdesivir will be approved within the coming months. The process, as I will show here very quickly, from raw material to active pharmaceutical ingredient or to API takes roughly 150 days. And given that it's a sterile product where manufacturing usually takes about 60 days, we've cut that process in half to just under a month. So we've made progress along the way. In the background, we're also working on an oral formulation that could be used particularly in countries and lower income developing countries to make it the distribution and access to it even easier, and shortening lead times where possible. In terms of access, clearly, we've been working on initially the compassionate use program, but quickly moving that into an expanded access effort, and in parallel, continuing to run the clinical trials, both in the US, Europe, and working very closely with the WHO on a number of protocols. And this is my last slide before handing over to Farah for Q&A. As I mentioned, we've made a commitment to donate the entire supply of Remdesivir that is currently being produced. And we're working with international groups such as the WHO, UNICEF, and the Bill and Melinda Gates Foundation to work on a sort of a quota system that is transparent and also on distribution channels, particularly for developing countries, so that we can make the drug available when and where needed quite rapidly. So with that, I will pause and hand over to Farah.
Thank you very much, Muhieddine. That's really exciting news, actually, to see such fast progress. I would like to take a minute to thank all of our speakers. I personally learned a lot. I hope that our audience has as well. I realized that there's just 4 more minutes on from our planned time. So I would basically suggest that we open the questions to the audience.
Yes, thank you. Let me jump in here. We were trying to answer most of the questions by text, so thank you so much, Ammar, and as well as Dr. Salwan. They answered some questions. There is a question by Ammar to Muhieddine, and the question was, what is meant by clinical improvement?
I would have a question.
Clinical improvement, there are a number of... Can you hear me, George?
Yes, go ahead. Sorry, somebody jumped on the call. Yeah, go ahead.
So I can share the exact details from the New England Journal of Medicine. But basically, it means removal of intubation and oxygen levels in the lung and other parameters. Actually, there are quite a number of parameters. So we can share with you offline what that is if the individual has specific interest in the criteria.
Great. By the way, I did answer also someone. If you do need to contact the panelists, I'll clear it with them offline. So send me an email at george@lebnet.us, and I can also forward some of your questions. So thank you for that. Now there's a question from Jeffrey Maruna. It's can someone comment on the receptor characteristics of this virus and how that affects the spread techniques to combat, and what is meant for the next one? And I'm sorry, and what it means for the next one. Can anybody take that quickly?
Well, I'll chime in quickly, as much as I understand the question. So the human receptor is ACE2, which is the site of attachment of the virus within humans. And that is not what the virus used to bind with when it was in bats. And so there was a natural development process between when it was in bats till it arrived in humans. That path is not known yet. How did that happen? Because many, they say, it's a conspiracy theory. Some, they say, no, it was already in Wuhan in their labs, and then somebody leaked that in a way or another. But the path from the bat to the human is not understood. The other thing, which is theoretical at this point, is that those types of receptors like ACE2 could be over stimulated or abundant in certain types of patients, like hypertensive patients, for example, and Salwan probably know that better than I do. And so there was an initial observation that those patients with coronary heart disease or coronary heart disease risk factors, they were impacted the most. Still early to talk about this, but I think that's important also to highlight that if this was really a virus manufactured to harm human beings, you could have done it with a receptor other than ACE2. There could have been other mechanisms that you could not even detect, and it could have been even much more mortal in that specific cases. I don't know if others would like to talk about that.
Anybody else wants to add anything before we move to the next question? Okay, so there's also a sort of a question/statement by Dr. Raed Al-Nasser. Dr. Sal, you said you were going to take a shot at it. I don't know if you want to read it before and answer it.
So this question came from Dr. Raed Nasser, the president of San Diego chapter of National Arab American Medical Association. I'm also a pulmonologist, critical care physician. Just want to raise a point. We are learning every day something new about this disease. Other systems involvement are being noted and recognized now. These include cardiac, gastrointestinal, renal, neurologic system involvement, among others. Thank you, Dr. Riyad, for this comment. Because when I earlier said this is a uni-organ disease, I was referring to for the majority of patients that encounter this virus, it tends to be localized to the respiratory system and an immune response. We are certainly seeing in hospitalized patients, especially intensive care unit patients who are really sick, secondary involvement of multiple organs, including the ones you've highlighted. And that begs for more research and more science to try to understand that. Thank you for making that clarification.
Great, and I'm also unmuting Dr. Raed in case, I know he has another statement regarding Remdesivir. I don't know, Dr. Raed, if you want to jump in and maybe mention it, or we can just read it on your behalf. Sorry, you're not unmuting for some reason. Oh, there you are. Can you hear us?
Yes, thank you. Thank you, Raed. Thank you very much for hosting this webinar. It's really very informative, and all your guests are really wonderful people around here. No, it just it's actually more of a comment rather than a question. Again, we need more studies to figure out what is the dose for the Remdesivir and how long, because really we don't know. You might be treating Ebola and other virus infections before, but in particular for COVID-19, this is going to be a different question. So what is the dose, how long you're going to treat it, and so on. So there will be a lot of clinical trials before we make assumption that the treatment is out yet. So these are all legitimate questions for clinicians like me. So more of a comment, really. Thank you.
Great. Thank you so much. So I'll tell you what, we still have 2 more questions. Let's just end with those. And I apologize. I was going to open it up to the audience for more, but since we need to answer the ones that have been asked here, let's do it. Dr. AbiEzzi, there's a question from Ammar again about should a diabetic take an ACE inhibitor hold on taking these pills for a while?
So the current, so ACE inhibitors is a class of antihypertensive that works on the ACE2 receptor. And when you take these medication, you tend to have overproduction of these receptors, and it's thought in the lung tissue especially, these receptors are all over the organ systems of the body, but specifically with the lungs, it's thought that maybe this can have a harmful effect, or some other research suggests a protective effect. So the jury is out. So far, the guideline from the American Heart Association and other strong organizations, they are recommending that if somebody is on these medications, which are widely used, is to stay on the medicine and not stop it during this pandemic. It's not likely that it's going to make a difference in terms of your capturing the disease, the COVID-19 disease, or dying from it. And the potential harm probably outweighs the potential benefit, because most of these people that actually die from cardiovascular collapse, not lung collapse, because we can put you on a ventilator, we can give you a lung assist, but it's usually the heart that's working harder and harder. If you take these medicines away, the heart is going to be subjected to higher pressures and will be subjected to stressors that eventually lead to cardiac arrest. So the answer is no, don't stop your medicines without talking to your doctor.
Great, thank you so much. There's another question by Charles Haj. It looks like that COVID-19 attacks mostly the lungs in children, and children seem to be safe. Is it possible that a tuberculosis vaccine might have a positive outcome too? This is by Judith Haj, actually, who asked the question. Anybody answer that?
I mean, there were anecdotal reports that show that perhaps those who have been immunized for tuberculosis are much more immune. I don't think, as Salwan mentioned, the jury is completely out on many of those questions.
Okay, great. Okay, so one more. Are albuterol inhalers harmful to take if someone is suspected of being infected? Sal, do you want to take that? Sorry.
Yeah, sure. So this refers to steroid inhalers, which are predominantly used by asthmatics or people with COPD. And the advice is not to stop your steroid inhaler. Steroids do weaken the immune system locally at the lung or respiratory tract level, and does increase your risk slightly for like pneumonia. But in this situation, it's probably the benefit of the inhaler outweighs its potential harm, and you should not stop using it. However, in the hospital setting, corticosteroids or steroids were used to treat these COVID-19 patients in China and then later on in Europe and the United States. And the jury on that is out that in general, corticosteroids given by mouth or intravenously tend to be more harmful than beneficial. So there we're again speaking predominantly to patients who don't really need them because of their asthmatic. They're being tried as a treatment for COVID-19. And in the United States, at least, it's not recommended that we use steroids to treat COVID-19 as of today. But if you are an asthmatic on an inhaler, you should continue to use your steroid inhaler.
This is excellent. And with that, we're going to end the session. Thank you all so much. I don't know if you have a last thing you want to say, Farah. This is your show.
Again, thank you everybody, the speakers as well as the audience for joining us. I hope you have learned new things today, and hopefully we'll have more of those to come in the future. Thank you again.
Thank you.
Excellent.
Thank you, Farah. Thank you, Ammar and Muhieddine. And thank you, George and LebNet community for hosting us and inviting us to this.
Stay safe.
No, thank you. Thank you to the panelists. Thank you for doing this. Appreciate it, everyone.
Take care.
Okay. We're going to be announcing another webinar for April 30th. It's more VCs and investments. Check your emails. Thank you.
All right.
Thank you.
Thanks.
Bye.